Hello everyone, I'm here to talk about my study. The purple acid phosphatase PAT26 is essential for efficient nucleotide metabolism in the vacuum of arabidopsis. I'm Liu. I'm a PhD student from Iowa State University. I work with Professor Gustavo McIntosh. So our lab work with RNA degradation in the vacuum of arabidopsis, and previously our lab has characterized the T2 ribonuclease RS2 as a key player in the RNA degradation of the vacuum. And we found that the impairment of RS2 stopped the RNA degradation in the vacuum. And the impairment of RS2 also increased the activation of autophagy inside the cell, and the activation of autophagy brings more RNA accumulated in the vacuum. In one of our collaborators, they published a paper suggesting that the impairment of RS2 seems to decrease the level of nucleotide in the cytosol, and the decrease of nucleotide level in the cytosol further activate autophagy through TOR complex signaling. So it seems that the RS2 dependent RNA degradation in the vacuum plays some role in regulating the nucleotide level in the cytosol. However, it has been published a couple years ago suggests that only nucleoside can be transported out of vacuum into the cytosol through the vacuolar transporter N1. So it is highly possible that the degradation product of the RNA inside the vacuum will be converted to nucleoside, and this will be part of the nucleotide substrate pathway. However, how the nucleoside is generated from the RNA degradation in the vacuum remains largely unknown. And on the other hand, the catalytic mechanism of RNase2 in the vacuum is also has not been studied in detail before. So in my study here, I propose that the vacuolar phosphatase PAT26 is a major phosphatase inside a vacuum that produces nucleoside from 3' nucleoside monophosphate. So 3' nucleoside monophosphate is the final product from the RS2 dependent RNA degradation. And the RS2 actually cleave RNA in 2 consecutive steps, so which produce 2' 3' cyclic nucleoside monophosphate as the intermediate product, and 3' nucleoside monophosphate as the final product. So in other words, PAT26 is the downstream effector of RS2 dependent RNA degradation in the vacuum, and both of these proteins play critical role in regulating nucleotide substrate pathway of the cell. Okay, so now we want to know whether PAT26 play critical role in phosphatase activity of the vacuum of the cell. So to test that, we use acid phosphatase assay to test the phosphatase activity in total soluble fraction and also the vacuum fraction. And we collect these 2 cell fractions from both wild type and PAT26 knockout plants. And you can see that in total soluble fraction, the loss of PAT26 decreased the activity up to 60%. And in the vacuum fraction, the loss of PAT26 decreased more, even up to 80% of the activity. So this suggests that PAT26 is actually the major contributor to phosphatase activity inside the vacuum. And we propose that PAT26 is the downstream downstream effector of RS2 dependent RNA degradation. So we expect to see that the knockout of PAT26 upregulate the level of basal autophagy in the cell. And here we use MDC staining to visualize the oligosome inside the cell. And after we do the quantification of the oligosome, we found that the number of oligosome in the cell of PAT26 knockout is quite similar to that of the RS2-2 cell, and both of them showed a higher level of oligosome number in the cell compared to that of the wild type, so suggesting that PAT26 has an upregulated level of basal autophagy, which is a phenotype similar to that of the RS2-2 cell. And in our collaborators' paper, they show that the treatment of 50 micromolar of ENC decreased the autophagy activity in the cell of in the plant tissue of from RS2-2 plant. So we also want to know that if PAT26 is a downstream effector of RS2 dependent RNA degradation, the treatment of 50 micromolar of ENC is expected to also decrease the autophagy activity in the PAT26 knockout plants. So we also here use the MDC standing to visualize autophagal zone inside the cell. And our data show that the treatment of ENC decreased the autophagal zone number in the RS2-2 cells. And also, we also see the same effect of the ENC treatment in the PAT26 knockout cells. So in both the mutant RS2-2 and PAT26 knockout, the treatment of 50 micromolar of ENC reduced the autophagal zone number effectively. So the data here suggests that support our hypothesis that PAT26 is the downstream effector of RS2 dependent RNA degradation. But we still want to know whether the catalytic mechanism of RS2 inside the vacuum. And through studying the literature, we propose that RS2 cleave RNA into consecutive steps. So in the first step, RS2 produce 2' 3' cyclic nucleoside monophosphate from RNA degradation. And in the second step, we propose that RS2 also carry this catalytic activity to produce 3' nucleoside monophosphate. So to test this hypothesis, we express arabidopsis RS2 with the HA tag at the C terminal. We express this recombinant protein in the yeast culture, and we purify this recombinant protein and want to know about whether our purified RS2 HA still has ribonuclease activity or not. So we incubate our recombinant protein with total RNA extracted from arabidopsis, and our in vitro RNA degradation show that our recombinant protein RS2HA did has ribonuclease activity at 25 degrees. So the incubation of the recombinant protein with total RNA suggests that the total RNA has been cut thoroughly at 25 degrees after 20 minutes of reaction. So next, we set up another RNA degradation assay, in vitro RNA degradation assay by using the polyadenylenic acid, or poly-A to be short, as a substrate. And we set up the in vitro assay at pH 6 and 25 degrees to mimic the environment inside the vacuum. So as we talked about this before, we expect to see two different products. The 2' 3' cyclic AMP, which is the intermediate product, and the 3' AMP to be the theoretical final product. And we first use the commercial RNA to test our assay. And as it has been published decades ago, we knew that RNA only degrade poly-A into 2' 3' cyclic AMP. And this is the same thing we see here in our assay. So after 20 minutes of treatment, most of the degradation product detected by the LC mass is 2' 3' cyclic AMP. And this result also suggests that there's no spontaneous conversion between the 2' 3' cyclic AMP and the 3' AMP. And next, we set up the same in vitro assay with the recombinant protein we have, the RS2HA. And again, we use LC mass to determine the level of 2' 3' cyclic AMP and the level of 3' AMP. We also want to know about the ratio between these two degradation products. So here is our result. It is quite interesting. As expected to see, LC mass detect the presence of 2' 3' cyclic AMP. And it also detects the presence of 3' AMP. And quite interestingly, the concentration of 3' AMP and the concentration of 2' 3' CMP is quite similar throughout the reaction. So which means that these two degradation products keep in the ratio of 1:1 throughout the reaction. So this suggests that the recombinant RS2HA has the activity to generate 3' nucleoside monophosphate as a final product, but this is actually a low efficiency process. So to be sure, in conclusion, our data suggests that RNase2 cleaves RNA in 2 consecutive steps. In the first step, the 2' 3' cyclic nucleoside monophosphate is produced, and from this intermediate product, the 3' nucleoside monophosphate is produced as the final product. But the production of 3' nucleoside monophosphate could be a relatively low kinetic efficiency. And second, our data suggests that PAC26 is a major contributor to the acid phosphatase activity in the vacuum. So it also suggests that PAC26 could be the vacuolar phosphatase that convert 3' nucleoside monophosphate into the nucleoside. And the mutation of PAC26 results in an increase in basal autophagy. So this is a phenotype that can be rescued by 15 micromolar of intensive treatment. So this suggests that PAC26, just like RNase2, is part of the nucleotide substrate pathway of the cell. And also, our data support our proposal that PAC26 work as the downstream activity of RNase2. So they produce nucleoside from vacuolar RNA degradation, which is again part of the nucleotide substrate pathway of the cell. And for our data here, we propose that the phosphatase activity of PAC26 is actually capable of promoting RNase2's catalytic activity in producing 3' nucleoside monophosphate through removing the final product continuously. So in other words, we propose that RNase2 actually requires PAC26 to increase its activity in degrading RNA inside the vacuum. So this is the end of my talk. My study has been supported by people working at Gustavo McIntosh lab and also the people working in Professor Bashan's lab. My work has been supported by the grant from National Science Foundation. So please let me know if you have any question.